Hello, welcome to video 28 for Mr. McGee's 9th grade biology class at Arizona School for the Arts. Welcome back. It's been a long time, ladies and gentlemen, long time. Today's video is about taxonomy and how to create a phylogenetic tree. Let's begin with the word taxonomy. Taxonomy is the scientific process of putting organisms into groups. Generally, organisms are grouped together based on the characteristics they share. In the 19th century, Charles Darwin proposed that taxonomy should reflect the evolutionary history of life on Earth. In other words, Darwin suggested that organisms should be placed into a family tree of all life. So taxonomy reflects not just similarities between animals, but also how they are related to each other through common ancestry. When taxonomy is represented as a family tree, it's called a phylogeny. The phylogenetic tree that includes every living thing on Earth has three major branches or domains. These are the eubacteria, which are small unicellular prokaryotes, the archaebacteria, which are similar to the eubacteria, but we tend to find them in really extreme environments like hot places, salty places, dry places, acidic places, you name it. Anywhere that seems like it's unforgiving for life, that's where they'll be. And the third group is the eukaryota, which includes virtually every form of life that is visible to the naked eye, and then a whole lot more. Each domain is then further divided into kingdoms and phylums and classes and orders, families, genuses, and then species. A phylogeny can be considered a type of hypothesis about the evolution of life on Earth. So scientists use all kinds of data to create phylogenies. Anatomical data, as well as fossils, genetics, developmental science. They use all of these to produce what they think is the most accurate phylogeny for large groups of organisms. Because scientists constantly accumulate new and better information, it's reasonable to expect that these phylogenies are going to change from time to time. For example, until recently, scientists hypothesized that the ancestors of whales were a type of carnivore, but new evidence demonstrates that whales are actually more closely related to things like cows and pigs and other hoofed animals. When you look at a phylogeny, what you're looking at is a family tree. The root of the tree represents the common ancestor of the entire group, and the branches represent the descendants of that ancestor. The place where the phylogeny splits into two branches represents what's called a speciation event, where a single species evolves into two species. Likewise, you can use a phylogeny to understand which species are closely related by looking for places where the branches are connected. These points, which are called nodes, represent common ancestors that are shared by the smaller branches on the tree. Phylogenies are constructed by comparing the characteristics of the organisms we are interested in. This can be done with a variety of useful data from things like genetics and proteins and anatomy, etc. For this example, we'll create a phylogeny for three imaginary organisms. They are the Forsmo, the Hector, and the Ragaragi. By looking at the similarities and differences between these three organisms, we may be able to determine how they are related. We can start by collecting some data about each organism. First, we notice that all three organisms have a one-way digestive tube with a mouth at one end and an anus at the other. Likewise, they all have two legs, fur, bilateral symmetry. In order to create a family tree, we have to look for some differences. For example, all three have eyes, but the Ragaragi and the Hector have only one eye each, and the Forsmo has two. Also, the Hector appears to lack horns, and both the Forsmo and the Ragaragi have adorable rounded tummies. Oh, look at those little tum-tums! Look at... When we compile this data into a table, it's easy to see a pattern. The Hector has more similarities with the Ragaragi than the Forsmo, and the Forsmo has more in common with the Ragaragi than the Hector. There's only one way we can arrange these organisms to reflect this pattern. Notice that the Forsmo and the Hector are far apart because they share the fewest similarities, while the Ragaragi is close to both. reflecting the number of similarities it shares with both species. But how do we know how to connect the branches of this tree? We need to review the characteristics that all three of these organisms share. Remember, they all have the same digestive tract, 2 legs, fur, eyes, and bilateral symmetry. These features represent the characteristics that their common ancestor would have had as well. Only the Forsmo and the Ragaragi have horns and rounded tummies. Look at those little tummies, so cute! These features represent more recent adaptations that only the common ancestor of the Forsmo and the Ragaragi would have. Finally, since only the Forsmo has 2 eyes, we can conclude that this feature represents the newest adaptation to evolve. So our hypothetical family tree would look like this. If these organisms were real, we could then test our hypothesis by comparing other anatomical features, or by looking at the similarities and differences in important genes or proteins. If we see the same pattern in the data, then we can be fairly confident that our hypothesis represents the real evolutionary history of these organisms. But wait! I know what you're thinking. If the Forsmo has 2 eyes, doesn't that mean that it's the most evolved? Now that is a scary thought. And you know, children, there's only one way to find out. The next time you see a Forsmo, try poking out one of its eyes, just to see what happens. Be sure to use something sharp, like a fork or a pointed stick, and remember to always wash your hands after mutilation. That's all for now, and as always, here's your bunny of the day.